Right now for the second half of photosynthesis. If you notice during the first half, we did use water, we used some light energy, oxygen was produced, but we haven't used carbon dioxide and no sugar has been made. So that's what happens in the second half of photosynthesis. To start the second half of photosynthesis, we're going to call that the light-independent reactions, and it begins with carbon dioxide that has gotten into the plant cells by diffusion from the outside in the atmosphere, comes in through little holes in the bottoms of the leaves called stomata and diffuses into each of the cells. Okay, carbon dioxide is going to combine with another chemical called RUBP. Stands for ribulose bisphosphate. You don't need to remember the name, but just realize that carbon dioxide comes in and combines. Now, to get this reaction to happen, it takes an enzyme, but this is not the most efficient enzyme, so notice I've drawn it with a pretty crummy looking mouth there. The active site fits carbon dioxide. It's not a great enzyme. It's not super fast. In fact, there has to be fairly high concentrations of carbon dioxide before this enzyme will work. The name of this enzyme is ribulose bisphosphate carboxylase, or RUBP carboxylase, but a lot of people give it the nickname Rubisco. It's a very abundant enzyme on the planet because of all the plants, and it's an inefficient enzyme, so plants have a lot of it to try to get this to work. Okay, when carbon dioxide and RUBP enter the active site of Rubisco, it combines those together to make a chemical called PGA. This chemical, now we can modify the structure of it by adding some energy to it. We're going to use the energy from, you guessed it, say ATP, that was just created during the light-dependent reactions. That leaves us with ADP molecules that can go back and be created again. The next step is also to modify the shape. We're going to use the other chemical that we produce having energy, NADPH, and that is going to leave NADP for us. Of course, each of these steps is accomplished by enzymes to do this work, and that leaves us with what's really an amazing thing again, is it recycles the original RUBP. Now the name of this cycle is called the Calvin cycle, or people also call it the light-independent reactions. Now, if you notice though, we can't add carbon dioxide to a molecule and end up with the molecule being the same size as it was to begin with. So each time this cycle goes around, we have to remove some atoms to regenerate the RUBP. So if we remove a carbon, a couple of hydrogens, and an oxygen each time, that's going to work. Once we've removed 3 of them, we create a chemical called G3P. If we go 3 times around the cycle, we can combine those 2 chemicals to make, well, if we've gone 6 times around altogether, we'll have 6 carbons, 12 hydrogens, and 6 oxygens, and that makes, you got it, glucose, everybody's favorite sugar. Now actually the glucose can then be changed. We can modify the glucose. Say it can be changed by, say, combining glucose molecules together. We can make starch for the plant. Or we could take the glucose, modify the structure of the sugars, and we can create other sugars like sucrose in like sugar cane and sugar beets. We can make fructose in the fruit. We can make all kinds of carbohydrates, cellulose for the cell walls. We can even do, by adding nitrogen, we can create proteins, and we can remove some of the oxygens and end up producing oils or lipids. So all these biochemicals began with glucose. That's produced during the Calvin cycle, the second half of photosynthesis. What a miraculous thing this is.